hit record, and then I am going to start the PowerPoint. So here we go. So chapter 1, you can probably go ahead and assume that it's kind of the introduction to our course, introduction to microbiology. So of course, you want to know what microbiology is. So our class is all about the study of microorganisms. So living things that are so small that they are invisible to the naked eye. That is basically what our class is all about. Okay, so we are studying the microorganisms on the planet, but our focus is very much on microorganisms that cause diseases in humans. So Dell Tech actually offers 2 different microbiology courses. Our class is really more what a lot of schools call medical microbiology, because there's lots of things in microbiology that we don't cover in our class, or we cover it a little teeny tiny bit instead of, going into a lot of detail. And that's because our class is very much more focused on human health and wellness. Okay, so this is the microbiology that people interested in medicine and healthcare take. The other microbiology is for, biology majors, people who want to do like research, become scientists, that kind of stuff. So of course you want to know what microbiology is, because that's the class you're in. So something that many of you probably don't know a lot about, unless you've had a general biology class, or if you took, biology somewhat recently in high school, is this thing called taxonomy. Now, taxonomy is a naming system that is used in biology to identify and classify living things. So I'm going to give you the categories here in a second, but the best way to think about taxonomy is just think about it in terms of an organization system, okay? Like if you had a collection, of like coins or stamps or baseball cards or whatever, you probably have some way of organizing it, right? So you might have it like by date, you might have it by value, you might have it by, how important it is, whatever. So taxonomy is kind of the same thing, but instead of organizing like coins or baseball cards, you're organizing life on Earth, okay? Now, you have to remember that the stuff that we talk about in our class is stuff that has been on the planet for billions of years, but people didn't know anything about them until the last couple, 100, 200 or 300 years. So it's kind of weird with microbiology because, when you think about living things on Earth, right? So you think about like plants and animals, okay? Humans have known about dogs and cats and horses and pigs and trees and bushes and flowers. We've known about those, like the whole existence of mankind, right? So when humans started to develop language, right, they developed words for the things that they could see, right? So like an apple tree, a maple tree, a dog, a cat, a lion, right? These things got names, okay? It started way back with, the ancient Egypt and ancient Greek. A lot of things were based off of Latin root words, and then you get to Romantic languages, and then you get to other languages like Chinese and English and all that. So things like that, macroscopic things that we can see. You go outside, you can see a bird, you can see whatever, a bee, you can see a flower, like you can see these things. So they have names, right? So then when bacteria, primarily bacteria, were first discovered, right? Nobody knew what the heck to name them, right? Because even though the bacteria were here first, humans didn't start discovering them and naming them until a couple hundred years ago, right? So it's like, what do we name it, right? Because there's no Latin word for a particular kind of bacteria, right? Not like there is for dog and cat. So when these scientists started discovering these different types of bacteria, they did what any person would do, right? They named it after themselves. They named it after what it looks like under the microscope. They named it after where it was found, right? And that's really how bacteria started to get named. Okay, and I'll give you some names here in a second. All right. So now, building on to this whole organization scheme, there's a message in the chat box. I think we were on a break, I think is why you were confused about it. And then I just now saw this message in the chat box too. Yes, I am recording. So anytime I'm doing a PowerPoint, we will be recording it. All right, so getting more into taxonomy and organizing all these things. In order to organize them, we need categories, and the categories are domain, kingdom, phylum, class, order, family, genus, and species. Now, when I took biology in high school, domain was actually not so much a thing. It was pretty new. So I learned a little saying from my biology teacher in high school, which is King Philip came over for good spaghetti. And that's how you can remember the categories, because there will be a question on the test where I ask you, what is the correct order of the taxonomic categories? So if you remember King Philip came over for good spaghetti, right, you should hopefully be able to get the question right, because the correct order is kingdom, phylum, class, order, family, genus, species. All right. And then we have this thing called the scientific name. The scientific name is the combination of genus and species. Okay. Now, just real quick, I'm going to give you an example of how we use these categories with two things that you should know about, because I really feel like you need an example before you can apply this to bacteria, essentially. So we're going to take a human and a dog, okay? We're not going to worry about domain yet. I'll get to domain in a bit here. So we're actually going to start with kingdom, all right? So a human, right, is a living thing, and a dog is a living thing. What type of living thing? They are both animals. So humans and dogs are in the same kingdom, kingdom Animalia, okay? Now, by the way, you don't have to write this down. You're not going to be tested on like the taxonomy of a human or a dog, okay? So I don't want you to think you have to write this down. It's just an example to help you understand it better, okay? So humans and dogs, they're both in kingdom Animalia, okay? Next is phylum. The phylum is Chordata. Chordata is referring to the spinal cord. So humans have a spinal cord, dogs have a spinal cord, so they are in the same phylum, okay? Now, there are animals that don't have a spinal cord, right? One really good example are like insects and spiders. So insects and spiders are animals, but they don't have a spinal cord. So they're in the same kingdom, but not in the same phylum. So we're basically getting more and more specific as we go. So next is class. Humans are in class Mammalia because we are mammals. We have hair and we produce milk for our young. Well, at least females can do that. Well, guess what? Dogs, same thing. All right. So humans and dogs are both mammals. So they are both in class Mammalia. Okay. So we just got rid of fish and reptiles and birds, and it's just mammals. All right. Now, order is when the two diverge. Okay. So humans are in order primates because we are primates. What makes us primates is we have an opposable thumb. Okay. This allows us to do a lot, right? And I'm not just talking about text messaging or typing or playing video games, right? Things like, you know, making tools and making weapons and building shelters, right? And climbing trees. That's what these are for. They're super important to a primate, okay? Now, dogs don't have this, right? So dogs are in order carnivora. So this is, like I said, this is where we get into two different categories, right? So humans and dogs, we're both mammals. We both have a spinal cord. We're both animals, but we're in different orders, okay? So now we're going to be in different categories. Humans are in family Homidae, which means man or mankind, okay? And then dogs are in family Canidae, which means dog, literally the Latin word for dog, okay? Now for the genus and species, that's where we get the scientific name. So humans, our scientific name is Homo sapiens, and that loosely translates to common man or, you know, current man, that kind of thing. So like a human, like just a human. For dogs, it's Canis domesticus, which literally means domestic dog, right? So like a wolf is Canis lupus, because that's what a wolf is. So lupo or lupine means wolf instead of dog. So you might be wondering, where the heck is he going with this, right? So I give you those examples so you can understand how taxonomy works, because when Carolus Linnaeus, who's kind of the father of taxonomy, when he was figuring all this out and kind of putting, you know, pen to paper and creating this system, there were other scientists who were just starting to discover bacteria. So it's like, here's the manual on taxonomy, and then all of these discoveries that are being made, you know, it's like, great. Now what do we do? Right? Like we have this system and the system is in place and it's been doing pretty good. And now we have this whole new world of life that we didn't even know was there. It was there the whole time. We just didn't know it because we didn't have the technology to see it. Right? We didn't have microscopes. We didn't have microscopes that were that good until, you know, the early 1700s. So I am going to type in the chat box because, again, I've taught this class enough in Zoom to know that students are going to want me to type it, okay? So I'm going to type some scientific names for you here. Now, I'm not going to, technically, anytime you type a scientific name, it should be in italics, but I'm sure there's a way to do that. I'm not going to use up class time to figure that out. But I am going to capitalize the genus, and then the species is always lowercase. All right, here comes bacteria, scientific name number 1. Staphylococcus epidermidis. Okay. So if you've heard of the term staph or staph infection, okay, it might be this, it might not be this, it might be a different type of staph, right? So Staphylococcus refers to what it looks like under the microscope, which we'll talk about next Tuesday when we do chapter 4. Epidermidis refers to the skin, because that's where this bacteria is found. This bacteria is the bacteria that lives on your skin. So that's how it got its name. This is what I'm talking about when I'm talking about when bacteria started to be discovered, the scientists had to figure out what to name it. So they were like, well, how about Staphylococcus epidermidis, because it looks like this and it's on the skin. Boom, there's the name. So then we get this one. Streptococcus pyogenes. So you're like, okay, well, I see Staphylococcus and Streptococcus, so I know there's some sort of similarity there, and you're right. We'll talk about it, like I said, next Tuesday. All right. So strep, like strep throat, right? This is the organism that causes strep throat. Streptococcus pyogenes. So the species name is kind of gross, but it's, you know, this class is gross. I mean, to be honest with you, everything we talk about is, you know, it's pretty gross. Pyo means pus and genes means to generate. So this bacteria causes the body to generate pus because this bacteria kills your white blood cells. And that's what pus is. So if you have like an abscess, or if you have some kind of wound or some kind of sore and you cut it or you squeeze it and pus comes out, right? That pus is dead white blood cells that have sacrificed themselves so that your body won't die, okay? Streptococcus pyogenes is one of the most dangerous bacteria that we know of. And you might think to yourself, but you're just talking about strep throat. I didn't think that was that big of a deal. Well, not in 21st century America. It's not that big a deal, okay? But I am telling you that lots of people die from this bacteria every day. It might not be in the United States, but somewhere. And there's lots of other diseases that this bacteria causes besides just strep throat. All right, the last one I'm going to give you here is a doozy, and spelling it right is challenging too. I think, I don't know if that's right. I thought it was right, but there we go, got it. Escherichia coli. So otherwise known as E. coli. I'm sure you've all heard of E. coli before, right? So we abbreviated E. coli just like we abbreviated staph and strep, because nobody wants to be going around saying Escherichia coli and Streptococcus pyogenes, okay? So Escherichia coli, the first part, the genus, was named after the person who discovered it. So literally, the guy's last name was Escherichia. And he said, well, I'm naming this bacteria after me. And that's what they did. A lot of bacteria are named after the person that discovered it. It's kind of a scientist's way of living forever. They're like, well, I may die someday, but at least this bacteria will always be here on Earth, and it's named after me. And then coli refers to the colon, because this bacteria lives in your large intestine. And another word for your large intestine is your colon. So E. coli bacteria is the bacteria that lives in your colon. It's in your fecal material. And the scientist decided to name that after himself. That's not something I would want to name after myself, but that's what he did. So that's how it got its name. So the taxonomy of bacteria is, you know, weird. That's the best way to describe it. It's a hodgepodge of things. It could be what it looks like, it could be where it was discovered, it could be who discovered it, all kinds of stuff. So that's why the names are so weird. There's a lot of really weird, hard to pronounce names in this class. And that's okay. You're not taking an oral exam. You don't have to pronounce these things, right? You just have to be able to pick the right answer on a multiple choice test. So just get kind of comfortable with some of these, wacky names. All right. And then, as you saw with like strep pyogenes, right? Oftentimes, the name of the disease ends up being very closely related, if not the same, as the name of the bacteria. So Streptococcus pyogenes causes strep throat. That's how it got its name. And I think that'll be hopefully useful as you're studying, because you can say, okay, this one sounds like this disease because it causes this disease. All right, so who are we talking about in our class? I'm going to say probably about 85 to 90% of everything we do in here is bacteria. Okay, then we will do quite a bit with viruses. Okay, viruses is not our focus, but it's definitely kind of like second place. All right, then we got some of these other weirdos that we'll talk about a little bit here and there. So bacteria fit into actually two different kingdoms as far as taxonomy is concerned. It used to be one, but now there's two of them. We have kingdom bacteria and kingdom archaea. Kingdom bacteria, that's the one that we really are talking about. Okay, kingdom archaea is like the ancient weird bacteria that lived like at the ocean floor, bacteria that lived in like deep caves, bacteria that live deep in the earth. There's even bacteria that can withstand living in volcanoes or like in geysers, ocean vents, just the crazy, weird, unusual bacteria. That's kingdom archaea. Okay, archaea means ancient or archaic. So they're the ancient bacteria. Kingdom bacteria is just the normal, regular, everyday bacteria that we come into contact with all the time. It's very important to understand that bacteria are everywhere all the time. They're on you, they're in you, they're on every surface, they're in your water, they're in your food, they're everywhere. And it's fine. That's living on Earth. That's the reality of living on Earth, okay? Because the stuff that hurts and kills bacteria is the same stuff that hurts and kills us, right? You can't live in boiling water. You can't live in bleach. You can't live in UV radiation, right? So we're just surrounded by bacteria all the time. It's in the water, it's in the air, it's on the surfaces, it's on us, it's in us. It's just, that's the reality and you have to get over it. I mean, I have a story about it, but I'll save it for later because I don't want to hold us up here tonight on the first night. But I will tell you my bacteria story eventually, I promise. All right, and then if you skip to the end of this little bulleted list, we have viruses. Viruses are terrible, right? Because COVID is a virus, so we all just have learned to just hate viruses. Viruses are weird because viruses are not made up of cells the way everything else on this list is made up of cells. So there's really no actual virus kingdom. Viruses aren't really named that way. I mean, they try. Taxonomists try to put viruses into like these little taxonomic categories. It just never really works. So we'll just, there's a lot more about that in chapter 13. So when you get to chapter 13, hopefully in the next week or so, you'll learn more about that. All right, and then we have kingdom fungi or fungi. That's fungus, okay? So when I say fungus, you probably think of things like mold or mushrooms, okay? Well, yes, that's fungus. And there are some types of mold that can actually cause diseases in humans. We also have a single-celled type of fungus called yeast. And yeast is very important in microbiology because a lot of experiments and a lot of tests are done using yeast. But yeast can actually also cause a couple different diseases that we'll talk about later on in the class. Then we have kingdom protozoa and kingdom chromista. Now, until somewhat recently, these two groups were in a single group called kingdom protista. And kingdom protista was basically just the miscellaneous group. right? So if it's not an animal, if it's not a plant, if it's not a fungus, if it's not a bacteria, then we just put it into that kingdom. Well, that wasn't specific enough. So then taxonomists decided, well, we're going to do two groups. We're going to do protozoa and algae. And, you know, a lot of this depended on the DNA technology and super, super incredible, like, you know, $1 million microscopes to figure this stuff out. And what they found out is a lot of the things that we thought would be in kingdom protozoa were not. So now it gets a bit murky. All right. Now, this portion is not going to be a huge part of any test. I don't expect any of you to be, you know, taxonomists or anything like that. But just so you're aware, okay, because there are protozoans that cause diseases, and then there are certain types of parasites that cause diseases. And those parasites, you know, a lot of people thought would end up being in kingdom protozoa, and turns out they're in kingdom chromista with the algae. And it's just kind of how things ended up to be. But, you know, all you really need to know is when, you know, throughout the entire semester, if I tell you a certain thing is a parasite or a protozoan, just know that that is a parasite or a protozoan. I'm not going to go into crazy detail like which one is chromista, which one is protozoa, you know, just don't worry about that. It's just not that important, honestly. What's much more important is you know which diseases are bacterial and which diseases are viral. That is very important. That is very important to your future career, okay, is to know which diseases are caused by bacteria, which diseases are caused by a virus, and usually which one in particular, but we'll get there. Now, also within kingdom Animalia are things that we will, for now, we'll call them, you know, disease carriers. I'll put it like that. I don't want to give you the vocabulary word yet. We'll get there. But certain types of insects, certain types of parasitic worms, they are technically animals, but they can carry and transmit diseases to us. So we have to talk about those as well at various points in the semester. So like I said, you know, when I say microbes, I mean all of them, but really the focus is on bacteria. Bacteria is everywhere, okay? Everywhere on Earth. In the air, in the water, bacteria can live in ice, bacteria can live in volcanoes. It's everywhere. That's just how it goes, all right? Now, there's a lot of stuff about microorganisms that we really don't talk too much about in our class, such as their role in ecosystems, food chains, industrial applications like making food and beer and wine, genetic research, which I love that stuff, but we just don't talk a lot about it in our class. Our class is by far about diseases or pathogenic microorganisms. A pathogen is a disease-causing microorganism. So you definitely want to know the definition of pathogen. Pathogen is a very important word for our class. And it's also very important to remember this, okay? Less than 1% of all the bacteria that we know of make us sick, okay? That means that 99% of all the bacteria on Earth does nothing to us. It's just living here. It's just doing its thing, okay? But about 1% will make us sick and could potentially kill us, all right? And that is by far our focus for our class, is on human health and wellness, essentially. All right, so more about bacteria. Now, this is just kind of a list of the general characteristics. Chapter 4 will talk so much more about bacteria, okay? Tons and tons of stuff about bacteria in chapter 4. So bacteria is made up of prokaryotic cells. And as a matter of fact, the term prokaryote is kind of interchangeable with bacteria. So you can say bacteria, you can say prokaryote, because only bacteria have prokaryotic cells, okay? And bacteria is unicellular. That means that they are made up of one single cell. So bacteria are single-celled organisms. Now, bacteria come in a variety of shapes, but for the most part, we're going to focus on the three basic shapes. First, we have cocci, which is spherical shaped bacteria, so bacteria that's in the shape of like a ball. Bacilli, which are rod shaped bacteria, so bacteria that look more, like cylindrical, almost like a little piece of rice. And then spirilla or spirilli, you can kind of say it either way, is spiral shaped bacteria. So bacteria that kind of look like a curled shape or like a comma shape, okay? So for now, you want to know these three terms for sure. But then when we do chapter 4 next Tuesday, we're going to take these terms and we're going to greatly expand upon them. So you want to know the three basic shapes before you want to get into all the chapter 4 stuff, okay? So you have spherical shape, cocci, rod shape, bacilli, spiral shape, spirilla. Most bacteria are heterotrophic, and heterotrophic, it translates to feeding off of others. Hetero means other, troph means feeding. This just means bacteria eat. They eat food, okay? And for the most part, bacteria like the same stuff that we like to eat. Most bacteria, they like starchy foods. They like sugary foods. Really, the faster that a food goes bad, right? The faster it spoils, that's how much bacteria love it, right? So things like fresh fruits and vegetables, bread, milk, cheese, right? Bacteria love all that stuff, okay? That's why, milk and bread, like it goes bad, like in a week. It's like, it stinks and it's gross. It goes bad because literally the bacteria, and a lot of times mold too, but mostly bacteria, the bacteria gets in there, starts eating it. That allows the bacteria to divide, which is more bacteria, more bacteria, more bacteria. And then once you get enough bacteria in there, they start to produce mostly acids and other chemicals. So when a bacteria eats like sugar, for instance, like sugar or starch, they will produce acids, and then the acids will break it down even further and basically make it spoil. So that's why food goes bad. It's because the bacteria is in there eating it and producing all these other chemicals. So we already talked about this. So we have kingdom bacteria and kingdom archaea. They are both made up of bacteria, just very different types of bacteria. All right, then we have fungus. So fungus is actually very much like us, okay? They are made up of eukaryotic cells. They are heterotrophic and they are multicellular, with the exception of yeast, okay? So the difference is fungus eats dead, decaying organic material. Animals do not, okay? Animals eat fresh food. Fungus eats like dead, rotting food, but we still eat. We're both still made up of eukaryotic cells. So I already mentioned yeast. Yeast is the one exception here. Yeast is a fungus, but it is unicellular or single cell. So the most common type of fungus is mold, and there are different types of pathogenic mold, which we will talk about later on, mostly in respiratory system, okay? And then not only can mold spores cause different types of respiratory and skin diseases, but there are people who are just allergic to mold. And allergies is a topic in chapter 19. So you gotta wait a little while before we get to that. So, remember, there's a difference between being allergic to something and then something actually causing a disease. Because you can be allergic to like pollen, but pollen doesn't cause a disease. You can be allergic to peanuts. That's not a disease. That's an allergy. There's similarities, but they are different things. And that is, pertinent to our class. So like I said, we used to have kingdom Protista. Now we have kingdom Protozoa and kingdom Promista. But they're all eukaryotic. They are all, for the most part, unicellular. And there are some that can cause diseases. So some that exist as pathogens or parasites. And we will talk about those. It's a very small little part of our class, but there are some very important diseases that you need to know that are caused by protozoa or parasites. And then viruses. So viruses are chapter 13. Chapter 13 is what you need to get to on your own. You have videos for it. You want to try to do it, within a week. If not, definitely within 2 weeks. Viruses are not made up of cells. Viruses are smaller than cells and they operate very differently. Some viruses have DNA, some viruses have RNA, some viruses have both. And then viruses are surrounded by a protein coat. So they don't have a cell membrane, they have a protein coat. Now, here is something important, although it's only 1 point, but still, okay? Now, there will be a question on the test that is a fill-in-the-blank question. The question is this: are viruses living things? The answer is whatever you think it is, okay? Now, if you ask me, if you say to me, Dr. Line, are viruses living things? I'm going to say no. I do not believe that viruses are living things because viruses are not made up of cells. And the way that I define life on Earth is something made up of cells. Now, that is a perfectly reasonable and accurate answer. Now, you may go up to somebody else and they might be Dr. So-and-so with like 10 different PhDs. And they may say, yes, I believe that viruses are living things because this. All right. This is a great example of there is no one answer to this. All right. So on the test, you have to put an answer. If you leave it blank, you get no points. But I will have the question on the test: are viruses living things? And you can put yes, you can put no, you can say I'm not sure, you can say I don't know, you can say maybe. All right, put an answer. So you get 1 point. It's like a free point on the test, but you have to put an answer. You can't leave it blank. If you leave it blank, you don't get a point. All right, so don't leave it blank. But what I want you to do is I want you to, after you go through chapter 13, go through the PowerPoint, go through the videos, you decide for yourself. If you agree with me that they're not living things, great. If you disagree and you think they are living things, also great. If you're not sure, you're like, you know what, I see both sides of it and I just really don't know. I'm just really not sure. That's fine too. There's plenty of scientists with tons of PhDs who are not sure whether they're living things or not. It's just one of those things. It'll probably never get answered. It'll probably never be decided. Whether viruses are living things or not, we just don't know. They're just too darn different. But unlike bacteria, remember I told you, most bacteria on Earth is harmless. Viruses are almost always bad. Viruses are almost always pathogenic. Now, it might not be a human virus, but it's a virus that attacks something, because the way that viruses operate, the way that they function, is they hijack cells. They get into a cell, they take over the cell, and then they kill the cell. That's what viruses do. That's how they operate. I'm not going to say live their life because, all right. So most bacteria is harmless. There's only a small percentage of bacteria that can actually make us sick or make anybody sick. Most of the bacteria is just hanging out, just wants to eat food, make more bacteria, and live its life in peace and not be bleached to death and not be irradiated to death. All right. But viruses are almost always bad. And there's tons of more information in chapter 13. So have fun with that. And of course, you can bring your chapter 13 questions to lecture next Tuesday, lab next Thursday, and then the test. All right, so now we're going to get into some of the history of microbiology, which is fun. You know, I'll try not to spend too much time, but there's so much good stuff in here, so much good stuff. And I will have a question for the class. I won't spend nearly as much time on it as I normally do, even though it's super fun. All right, so it's widely accepted that prokaryotic cells came first because prokaryotic cells are much less advanced. They're much less complex than eukaryotic cells. So it just makes sense. Okay, there's this thing in science, it's called the principle of parsimony, which is if you can't explain something fully, then the most reasonable and logical explanation is the one that you go with. So what came first, the prokaryotic cell or the eukaryotic cell? There's no way that we can actually say definitively which one. But what makes the most sense is that prokaryotic cells came first because they are less complex. It just, why would the more complicated thing come first? It just doesn't make sense, you know? Like what, you know, why would, you know, the army tank come before the spear? Just doesn't make sense. Now, this is another great question. Who discovered microorganisms? Well, we don't know. So this is very much up for debate because you could say, okay, who discovered the cell, right? Or who discovered bacteria, right? So it's very hard to answer because you can say, all right, well, who first discovered the first microscopic thing versus who discovered the first microorganism, right? Totally up for debate. So the first scientist we're going to talk about is Robert Hooke. Now, Robert Hooke was very much a biologist, very much a serious scientist, and Robert Hooke is the person who coined the term the cell. So Robert Hooke had some okay microscopes. They weren't great, but they were okay, all right? And he was looking at things like plant cells and cork, like he would take pieces of cork and he would take leaves and he would look at them, okay? And when he looked at these things, he looked at them under the microscopes and he was like, well, these look like jail cells. Like they literally look like where we put criminals. And he thought, oh, I'm going to call this the cell. And that's how we got the term the cell. You know, like living things are made up of cells. Like that was Robert Hooke. He thought they looked like jail cells. And he was like, I'm just going to call these the cell. And he published all these papers and all this kind of stuff. Now, here's the thing about Robert Hooke, okay? He was very arrogant. He was very full of himself. And people don't really like that. You know, like we don't like it nowadays, and we didn't like it 400 years ago, right? So you have to, I mean, really, the scientific community hasn't changed a lot in terms of like this stuff. Like there are scientists who just think that they're so great and they're so smart and they're like the smartest people on earth, right? And they don't have a lot of friends, you know, because people don't like it. You walk in the room and you're like, oh, I'm so much smarter than everyone here. This is ridiculous how smart I am. People don't like that, right? And that's how Robert Hooke was. Like Robert Hooke legitimately thought he was like one of the smartest people on earth. And he let other people know that, right? Well, the other scientists in Europe, because this is all Europe, you know, They didn't like him at all. You know, they're like, oh, here comes Robert. Geez, what's he going to say tonight? You know? So he probably did discover a lot of the things that he said he discovered, but because he was not well liked, a lot of his stuff kind of got like pushed under the carpet. You know, like he would submit his papers to like, you know, University of blah, blah, whatever. And they'd be like, yeah, okay, whatever, Robert Hooke. Okay. So yes, he discovered a lot. He was very important in the world of biology, but they didn't like him so much. So he didn't really get a lot of credit that maybe he should have gotten. All right. So then we get to Anton von Leeuwenhoek. And yes, that's his actual name. Okay. Now, he was known as the best lens maker in the world. Okay. So von Leeuwenhoek was a haberdasher, which is someone who makes fabric or clothing. So what was happening with him is as he got older, it was getting harder and harder to see the thread and to thread the needle. If you've ever tried to thread a needle, okay, when you get older, it's hard to do that, right? So he was like, all right, if I'm going to stay in business, if I'm going to keep making these clothes and these fabrics and these curtains and rugs, whatever he was making, okay, he's like, I got to be able to see what I'm doing. So he would actually take pieces of glass and he would grind them down into lenses. And he would use these lenses and he would set up this little device and he would put lenses on top of lenses. He would stack them up like a stack of coins. And then when he would look through it, he could see it. Like it was huge, you know, the way he magnified these things. So he's going about his day, you know, making the thread and stuff. And he starts noticing other things under these microscopes that he's making. And he is basically given the title of father of microbiology because he pretty much is, you know, recognized as the first person to ever see bacteria. Because remember, bacteria is way, way tinier than the plant cells that Robert Hooke and other people were looking at, okay? Von Leeuwenhoek made the best lenses, which means he had the best microscopes of anybody. And there's actually this story where he was literally called to the court of Peter the Great in Russia because of how famous he was. And Peter the Great was like, let me see these lenses that everybody talks about. And he's like, here you go. And he's like, well, these are just plain lenses. You know, I heard you could see things that no one else can see on the whole planet, you know. And he's like, oh, those are my lenses. Nobody gets to see those. You know, and everybody was shocked. Everybody was like, this guy just said this to Peter the Great. Like, you don't say that to Peter the Great, you know. And von Leeuwenhoek stood his ground. Like, he was a stubborn old man at this point. And he was like, no, they're mine. I made them. They're mine. Nobody gets to see them but me. And that's why it took probably almost another 100 years for really good microscopes to get made, because von Leeuwenhoek refused to share lenses with anybody else. So nobody else could have these super awesome microscopes that von Leeuwenhoek could make. It's wild, wild stuff. And then the person who actually gets the credit with developing the first compound microscope, which is before all these other folks, was Zacharias Janssen. It's actually a father and son group, the Janssens. So they kind of furnished or fashioned the first microscopes, and then people like Robert Hooke and von Leeuwenhoek just kind of took it that extra steps. Now, chapter 3 is all about microscopes. It's a very short chapter. There's not a lot of questions on the test, but chapter 3 is the next chapter you'll want to do, and you're going to be doing that one on your own. So you definitely want to get chapter 3 done before next Tuesday, but it's super short. And obviously, we're going to talk about microscopes in lab on Thursday as well, because we have to do microscopes a lot in our class. Like microscopes are super important in microbiology. Okay, so now we get to this idea that life can spontaneously generate from non-living matter. So there's scientists who say yes, and there's scientists who say no, and they're trying to figure out, okay, can life just sporadically just come about? Just poof, there's life. Well, no, definitely, absolutely not. And the person who disproved the theory of spontaneous generation was Louis Pasteur. So if you've never heard of Louis Pasteur before, you're going to hear a lot about him in our class, because von Leeuwenhoek is the quote unquote father of microbiology. Louis Pasteur is like the father of modern microbiology, or like medical microbiology. Like it's one of those like kind of weird things. Like von Leeuwenhoek, he gets that title because he's the very first person ever to see bacteria, right? But Louis Pasteur was, I mean, von Leeuwenhoek, he was, a haberdasher. Louis Pasteur was like the scientist of his time. He was a biologist, a botanist, a chemist, a microbiologist. He did tons of stuff. And this is one of the things he's most famous for, is disproving the theory of spontaneous generation. Now, his experiment, very famous experiment, it's on page 8 if you want to take a look at it in the book, okay? And I put the year here. I will not test you on any years, okay? So on the test, there will not be any questions that say, in what year did so-and-so do this, okay? Just because I feel like as long as you know who did what, I feel like that's the more important thing. Like this isn't history class, right? If it was history class, then yeah, you got to know the years, okay? But I don't want you to be spending a lot of time memorizing years. It's just not, I just don't feel like that's as important as just knowing who did what, okay? All right, so let's talk a bit about Louis Pasteur. Like I said, he was a chemist, he was a biologist, he was a lot of things. He did a lot. And yes, he was French. He discovered that microorganisms can live on solids, in liquids, and in the air. So he's the person who first proved that bacteria lives in water, lives in the air. It's everywhere. Basically, it's everywhere. Louis Pasteur is the one who developed aseptic technique. Aseptic technique is very important. We are going to learn about it in lab. So you're actually going to be performing and using aseptic technique. Basically, it's just using the fact that heat will kill bacteria, and it's a way to prevent contamination, because you don't want to be spreading bacteria around the lab or around the hospital. That's not good. You're going to get people sick. And in today's modern society, you're going to get people sued. And it was also Louis Pasteur who figured out the mechanism behind alcohol fermentation. So, of course, humans had been fermenting things for, thousands of years before that. It did not take the cavemen very long to figure out that if you take some fruit and if you mash it up and if you let it sit for a while, and then if you drink the liquid, you will get drunk. Trust me, it didn't take humans long to figure out how to get drunk. We just didn't know how it worked. We just knew that if you take stuff made of sugar and you let it sit around in a bowl or in a jar, and then when it starts to kind of smell a little weird, you drink the liquid and you're drunk, okay? It was Louis Pasteur who proved that yeast have the ability to turn sugar into alcohol, and he called it alcohol fermentation. So that was also Pasteur. Pasteur also developed, well, it's literally named after him, pasteurization. So pasteurization is the process in which liquids are heated at lower temperatures for a longer period of time in order to kill all the bacteria. So this was done at first with beer and wine because, of course, a lot of bacteria can also perform alcohol fermentation. It's just yeast can do it a lot better and a lot quicker. But you don't want to have a bunch of beer or a bunch of wine that's full of yeast and full of bacteria, because that it can also make you sick too. I mean, it makes you drunk and it makes you sick. So through the process of pasteurization, the heating through of the beer or wine would kill all the organisms, but still the alcohol was there and people could still get drunk. They just wouldn't get sick. Then how about we apply that to milk? And that absolutely revolutionized the dairy industry. I mean, this is one of the reasons why people were able to make so many more people during this time period, the late 1800s, the early 1900s, because milk was a very important food for people, right? But up until that time, you know, you would milk the cow and you would have to drink that milk within a couple hours or it would go bad, right? And even if you kept it cold and even through the process of the invention of refrigeration and all that, you still need the pasteurization, because putting something in the fridge is only going to slow the bacteria down. But heating it up first will kill the bacteria. So you milk the cow, you heat up the milk, then you put it in an airtight container, and then you put that container in the fridge, right? Then your milk will last for a week instead of an hour. So yeah, revolutionized milk as they knew it then. Okay, now we have this part, and let me just check the time. All right, I'm doing okay. Like I said, I can't spend as much time on this as I would like to. All right, so my question for you, if you can think of something, great. If not, it's not the end of the world. Okay, so I want you to go back. I want you to go back to, let's say the year, let's see who knows their history, 1492, right? So the year 1492, there was like some stuff that happened, okay? We're not going to talk about that stuff because it's not history class. What I want you to think about is this, okay? You are just a regular, everyday person living in Europe in 1492. My question is, what makes people sick? So think about it for just a couple seconds. It is the year 1492. You're living in Europe in 1492. Doesn't even have to be Europe. It could be anywhere on Earth at this point. 1492, what makes people sick? Why do people get sick? Like physically ill and die. Why does that happen? Any ideas? If you're too shy, you can type something in the chat box. You can even send it to me privately. I want you to think a little here, because I've been doing an awful lot of talking tonight. Oh, I see the chat box coming up here. Let's see. All right, so I see a disease, I see traveling, I see chickenpox, okay, bad hygiene is a little better, we're gonna get there. So yeah, bad hygiene's pretty good, but we're not there yet.
Water contamination.
So another pretty good one, but we're not there yet. So somebody is saying about how, yeah, 1492, there is no such thing as microorganisms. There is no such thing as bacteria. There is no such thing as microscopes. There's medicine. Whether it does much, we'll talk about that. That's a chapter, I forget what chapter. We'll get there. So in the year 1492, why do people get sick? Why do people get physically ill and die from being sick? So sneezing, coughing, vomiting, diarrhea, get worse, die. Why did that happen in 1492? So I'm watching the chat box. I mean, they're not bad answers. It's just they're not 1492 answers. They're, you know, they're the things that you all should be thinking of. But we're talking 1492. I said we're not talking about that other history stuff. Oh no, they can cook their meat. It just like I said, it didn't take cavemen long to figure out how to get drunk. It did not take them long to figure out how to make a fire and cook their meat. No such thing as microbes. There's no such thing as clean and dirty. It's 1492. Everything's dirty. So you ready for the answer? In 1492, people got sick and died because God was punishing them for being evil, or a witch put a curse on them, or they were possessed by a demon. That's why people got sick in 1492, right? That is what humans thought for hundreds and hundreds of years, right? The reason that you're throwing up and having diarrhea and coughing and sneezing and coughing up blood and passing out and dying, it's because you made God angry, or you made a witch angry, or you're possessed by some sort of demon or spirit. Think about that. That was the truth for hundreds and hundreds of years. So then you get to the 1600s and the very early 1700s, but mostly the 1600s. And that's when we get more to a lot of what you were all saying about bad water, bad food, bad air. Okay. So by the 1600s, most people, except for maybe, some offshoots and maybe some, very low class levels or whatever, most people throughout the 1600s just thought that, you got some bad air in you, you got some bad water in you, they still weren't using the term bacteria, diseases, infections, right? There was this term very popular in the late 1600s. The term was miasma. And miasma was just the word that they used to kind of describe like the bad air, the bad water, the bad surroundings, right? So they would say, go get some fresh food, go get some fresh, clean water, go get some fresh air, get out of your house, go out to the country, right? Get out of the city, go to the country, get some fresh air, get some rest, get some sunshine. Like all those things like worked. So they thought that that's what it was, but that's not what it was, right? So then we get back to the PowerPoint, which talks about this thing called the germ theory. And even in the mid 1700s, a lot of scientists and doctors did not like the word germ, because it was mostly because they either didn't understand it, they didn't want people to think that's what it was. It was just a weird kind of time, I guess, in science, is, the mid to late 1700s. But more and more doctors and scientists were starting to believe that these microscopic organisms, these bacteria, could potentially be the things that make us sick, right? Now, of course, nowadays, I mean, a 5-year-old knows this nowadays, especially in the world of COVID, right? Everybody knows now. But, back then, it was very divided. It was a very divisive thing. It was, it was almost like a Republican Democrat thing, how it is nowadays, right? Back then, it was like germs or miasma. Is it just bad air, bad water, or is it an actual living thing that gets in you and makes you sick, right? So they go back and forth. What is it, right? Now, of course, Louis Pasteur was a huge, huge proponent of the germ theory. Louis Pasteur was like, look, I have shown you that these things live in the air. They live in the water. They're in our food. They're in the soil. They're on your skin. They're in your spit. They're in your feces. How can you not think that these are the things making you ill? That was Pasteur's view. All right. So then we get another person, Joseph Lister. And yes, Listerine is named after him. Okay. Now, the story of Joseph Lister is a good one. It's actually a really great one. I'm not going to get into too much detail, but basically, he was a young doctor. His father was a doctor. Probably his grandfather was a doctor. You know, he was from a very well-to-do family. He had plenty of money, went to the best medical school he could go to, right? And he was a young doctor, fresh out of medical school. He wanted to be a surgeon. He wanted to be like one of the world's greatest surgeons and all that kind of stuff. And he believed the germ theory so much that he is the first person to start disinfecting wounds and surgical equipment. Just imagine nowadays if a doctor does surgery and he's like, okay, take him away, bring in the next one, and he just starts doing surgery again and doesn't like wash his hands, doesn't clean the instruments. Like nowadays, just that whole hospital will be shut down. Guaranteed, shut down. And he's gone. He's fired, right? But back then, they didn't do that, right? So people would get infections and die, and they'd be like, well, they were going to die anyway before I did surgery, so not my fault, right? So it was Joseph Lister who tried to get people to do these things. Like, okay, before you do surgery, right? Wash your hands really good. Scrub the patient's skin really good, okay? Put the scalpel and the scissors and the sutures, put all that stuff in chemicals to kill the microorganisms and then do the surgery. And so many doctors were like, nah, you're wrong. It's not going to make a difference, right? And then what started happening is Joseph Lister's surgery patients didn't die of infections. They got better, right? So Joseph Lister had like the highest survival rate of his, patients of any doctor in the world. And they were like, oh, okay, maybe he's on to something, right? So that was pretty important. But the person who actually gets the credit is not Louis Pasteur. A lot of people think that it is because he really, really wanted to be the person who got this title as well. But the title goes to Robert Koch. So Robert Koch is the person who definitively proved the germ theory of disease. He proved that the disease anthrax is caused by a bacteria that was named Bacillus anthracis. So we'll talk a little bit more about Robert Koch when we do, pathogenicity and the process of disease. But basically, he just worked out a series of steps, experiments that became known as Koch's postulates. And it's basically how he proved that, diseases are caused by these microorganisms, this bacteria. So stuff that we can't see that is alive gets in us by a whole bunch of different ways, makes us sick, causes those problems, right? It was huge, huge, okay? Now, all this stuff was going on in Europe, right? In the United States, in the mid-1800s, the country was torn apart by the Civil War. So the United States, pretty much through the entire century was out of the loop, so to speak, okay? This is why in the early 1900s, the United States was not viewed very well by mostly Europe, I mean, you know, China and Japan, they didn't care, they kept to themselves, but really it's mostly Europe. You know, they thought America was like, you know, dumb, basically. So it wasn't until World War I that we started to produce munitions and we started to produce weapons and we supported, you know, certain groups in Europe. And then, of course, through that support, they were like, okay, maybe they're not so bad. And then, you know, World War II, of course, is when we got a lot of the respect that I think we still have today, but we won't get into the politics of all that either. But, you know, all these super important people in the world of microbiology, in the world of medicine, Louis Pasteur, Robert Koch, Joseph Lister, this was all Europe. You know, there wasn't a lot of science and medicine in the United States in the 1800s, mostly because of the political strife and because of the Civil War and then everything that came after the Civil War. You know, it really wasn't until after World War I and then certainly World War II that we became the world leader in science and medicine. It took